Okay, so far we have primarily focused on the anaerobic system, so the phosphagen system and glycolysis, which dominates the short duration, high intensity activity. But now we will take a look at aerobic ATP production, which we know is going to take over as exercise intensity decreases or duration increases. So, aerobic ATP production takes place in the mitochondria. And so, this system relies on the cooperation of two major processes. You have the citric acid cycle or the Krebs cycle, and the electron transport chain. And together, these two pathways complete the oxidation of carbohydrates, fats, and in some cases, proteins to produce ATP. So, when we first look at the citric acid cycle or the Krebs cycle, we see that when oxygen is present, pyruvate, so remember the end product of glycolysis. So, if we actually look at this picture right here, we see glycolysis has occurred. We are left with our end product, pyruvate. When oxygen is present, we see that it will be shuttled into the mitochondria and converted to acetyl CoA. Now, each glucose molecule is going to produce two acetyl CoA molecules. So the cycle is essentially going to run twice. And we know that each glucose molecule produces two acetyl CoA molecules because it's the two pyruvate molecules that we get at the end of glycolysis. So one pyruvate molecule gets shuttled to the mitochondria where it's converted to acetyl CoA, and then the other one is shuttled to the mitochondria to be converted to acetyl CoA. From there, what we will see is that acetyl CoA will combine with oxaloacetate to form citrate. So oxaloacetate, 4 carbon molecule, acetyl CoA, a 2 carbon molecule, joined together to form a 6 carbon citrate molecule. Now, through a series of steps, citrate is metabolized back into oxaloacetate, and along the way, we see that CO2 will be released, which is what we will exhale while we are performing physical activity. And probably the most important steps of the citric acid cycle is going to be where hydrogen atoms are stripped away and picked up by electron transport molecules. So your NAD plus and FAD forming those NADH and FADH2 molecules. And those molecules will now carry high energy electrons into the electron transport chain. And so it's here in the electron transport chain where electrons are passed along a series of protein complexes in the inner mitochondrial membrane, releasing energy at each step. So that energy is used to pump hydrogen ions across the membrane, creating a gradient. And then finally, those hydrogen ions will flow back through ATP synthase, driving the production of ATP from ADP. And this is referred to as oxidative phosphorylation. So the citric acid cycle, Krebs cycle, referred to as substrate phosphorylation. The electron transport chain is oxidative phosphorylation. And then at the end of the chain, the electrons will combine with hydrogen and oxygen to form water, which is why oxygen is essential for this process. Without oxygen, there is no aerobic metabolism. Now, aerobic metabolism produces way more ATP than the anaerobic pathways. So remember, the phosphagen system will produce 1 ATP. Glycolysis produces 4, but we had to use 2, so we only really get 2. Where aerobic metabolism is going to produce roughly 32 ATP per glucose molecule. But the downside is that it's much slower, which again is why it's the primary energy system for longer duration, lower intensity exercise like jogging or cycling, as opposed to sprinting or jumping. Okay, so now let's take a look at the citric acid cycle specifically. So again, when oxygen is available, pyruvate, end product of glycolysis, shuttled down to the mitochondria, where it's converted to acetyl-CoA. Now, remember, pyruvate is a 3-carbon molecule, while acetyl-CoA is a 2-carbon molecule. And so we see at this step, CO2 is actually formed and released. Now, acetyl-CoA is then combined with oxaloacetate, which is a 4-carbon molecule to form citrate, which is that 6-carbon molecule. Sorry, I forgot to point it out. So pyruvate converted to acetyl-CoA, and we have CO2 and formed and released. And then we have that acetyl-CoA, which is going to combine with oxaloacetate to form that 6-carbon citrate. And this is why sometimes it's referred to as the citric acid cycle as opposed to the Krebs cycle, which was named by the scientist. Now, from here, we see citrate goes through a series of enzyme-driven steps, ultimately being metabolized back into oxaloacetate to cycle through again. Now, during this process, there's 2 molecules of CO2 that are given off, again, that we will exhale, and 3 molecules of NADH, which we see here, here, and here. And then 1 molecule of FADH2 that are produced. These are the hydrogen and electron carriers that go on to fuel the electron transport chain. So they aren't considered a part of the Krebs cycle as far as the ATP produced because they are being transported to the electron transport chain. The only part that we see where we have this substrate phosphorylation is going to be when succinyl CoA is converted to succinate. We see that a GTP will form with the addition of an inorganic phosphate to GDP, and that GTP will donate the additional inorganic phosphate to an ADP, producing a single ATP. Now, because we only have a single ATP in that's generated from the cycle, but we have 2 acetyl-CoA molecules from that 1 glucose molecule, we get a total of 2 ATP produced during the Krebs cycle. So the main job of the citric acid cycle isn't actually to produce a ton of ATP directly, although a lot of individuals believe that's the case when they're first learning it. But in reality, we're only getting that 1 ATP per turn. Uh, it's more about, or it's more important to strip off the hydrogen atoms and electrons and load them onto the NAD plus and FAD molecules so that they can be transported to the electron transport chain where the bulk of ATP production actually takes place. So think of the Krebs cycle or the citric acid cycle as a hydrogen and electron harvesting system uh, that finishes breaking down the fuel and then sends the electrons to the electron transport chain for that large scale ATP production. Okay, so I want to pause for a second before we go to the electron transport chain because up until this point we focused mainly on carbohydrate metabolism, but during longer duration exercise, we also rely heavily on fats as well, and to a lesser extent, proteins in order to generate ATP. So, fats are going to be stored as triglycerides in the muscle and adipose tissue, and they are going to have a structure that looks something like this, where you have a glycerol backbone and then 3 fatty acids attached to that glycerol backbone. And before we can use fats for energy, triglycerides have to be broken down into that glycerol backbone and the separate fatty acids. And the fatty acids will be the major fuel source for muscle during exercise. They're going to undergo a process called beta oxidation, which takes place in the mitochondria. And this process chops or cleaves the fatty acid chain into 2 carbon segments. So if we look at probably another poor drawing, we see that these fatty acids are going to be made up of a hydrocarbon chain. And then we'll have hydrogens attached to this carbon chain. And then for the sake of time, I won't draw out all of the hydrogens, but you get the point. And what we see is that the typical fatty acid used for energy is going to be palmitic acid, which is going to include 16 of these carbon molecules in a chain. And so when we cleave or chop the fatty acid up into 2 carbon segments, we end up with 8 2 carbon segments with that palmitic acid example. And then they are converted into acetyl-CoA and enter the citric acid cycle. Now, glycerol can be converted into glucose in the liver, but it's not a major fuel source during exercise. So again, it's the individual fatty acid chains that are the primary source of energy from the triglyceride. Now, proteins are normally going to be spared for structural and functional purposes, but under certain conditions like prolonged exercise or periods of starvation or extended fasts, they can be broken down into amino acids and used for energy. And so amino acids can undergo deamination, which removes the nitrogen containing amino group. And we can also alternatively see that they can undergo transamination, which is where the amino group is simply transferred to another molecule to form a new amino acid. Both scenarios, that specific amino acid loses its amino group. And both of these processes require a coenzyme called PLP, which is derived from vitamin B6, which, side note, just fun fact, I guess, this means that if you have a deficiency in vitamin B6, this can impair protein metabolism. Now, under normal conditions, cells are going to prioritize carbohydrates and fats for energy and reserve amino acids for building and repairing tissues. Only in extreme cases will we use protein and their amino acids to generate ATP. So, just to sum all of that up, fats are going to provide the largest yield of ATP out of the macronutrients, but are mainly reserved during low intensity, long duration exercise. And then proteins can contribute, but they contribute very minimally under normal conditions. Okay, so continuing on with utilizing fat as an energy source, we see that first fatty acids have to be activated before they can be used. And so this happens in the cytoplasm where the fatty acid binds to coenzyme A, forming fatty acyl CoA. And this process is catalyzed by the enzyme acyl CoA synthetase. And this process, this activation step, costs 2 ATP. So we have an initial energy investment similar to the one we had with glycolysis. Now, once activated, the fatty acyl CoA is transported across the outer and inner mitochondrial membranes into the mitochondria where beta oxidation occurs. Now, inside the mitochondria, we have this repetitive sequence where we cleave off or chop up 2 carbon segments from the fatty acid chain or one at a time. And each 2 carbon fragment is going to be converted into acetyl CoA, which then enters the citric acid cycle. Now, an important detail is that for each chop, we actually get one NADH and one FADH molecule that is produced. So these electron carriers then enter the electron transport chain and will yield about 4 ATP per cycle. About 2.5 for the NADH and about 1.5 for the FADH 2. And this is even before the acetyl CoA goes through the Krebs cycle. We're already getting some of these hydrogen electron transport molecules flowing to the electron transport chain, which we know makes up the bulk of our ATP production. Now, the total amount of ATP we get depends on the length of the fatty acids, right? So if we're already getting 4 ATP for every time we chop the 16 carbon fatty acid or a 14 carbon or a 18 carbon fatty acid, we will get that additional 4 if it's, say, an 18 versus a 16. So we also see that the quantity of acetyl CoA that will enter the Krebs cycle will be different as well, right? So, for example, palmitic acid is that 16 carbon fatty acid. It's what is typically used for energy. And so, if it's a 16 carbon, it's going to yield 8 acetyl CoA molecules, because 16 carbons divided by 2, because we're chopping them up in groups of 2, we get 8 groups. Now, this is going to result in 7 of those chops, which produces 7 NADH and 7 FADH2 molecules, which together yield about 28 ATP, eventually through the oxidative phosphorylation going through the electron transport chain. Now, each of the 8 acetyl CoA molecules then enter the citric acid cycle, producing the additional NADH, the FADH2, and the GTP, which translates to ATP. And this is ultimately going to give us about 106 net ATP once we subtract the cost involved in these processes. Now, here, real quick, we are just taking a step out, since we talked about the different macronutrients and how they can be utilized for energy. And so, even though these 3 macronutrients are different chemically, they all eventually converge in a common pathway, which is being converted to acetyl CoA. So carbohydrates are broken down into glucose, which actually let me go. Broken down into glucose, which goes through glycolysis to form pyruvate, or in this image, pyruvic acid, and then pyruvate is converted to acetyl CoA. Fats are going to be broken down into fatty acids through lipolysis, and then those fatty acids undergo beta oxidation to eventually form acetyl CoA. And then proteins are broken down into amino acids, which can be converted into glucose, pyruvate, or directly into acetyl CoA, depending on the amino acid. And we also see that depending on the amino acid, it could enter the citric acid cycle at various points. So proteins are a little more complicated and can vary based on the amino acid. So once we have acetyl CoA, no matter which macronutrient it came from, it's going to enter the citric acid cycle, or the Krebs cycle, where it is fully oxidized. And this means all 3 macronutrients ultimately feed into the same energy producing pathway, just through various entry points. And we are finally back at the electron transport chain. So most of the video so far has been focused on the citric acid cycle, or the Krebs cycle, and just looking at how different micro or macronutrients are used or enter the Krebs cycle or citric acid cycle. Now, we know the electron transport chain is coupled, or I should say works in conjunction with the Krebs cycle. So the electron transport chain takes place in the inner mitochondrial membrane and is responsible for the majority of ATP produced during aerobic metabolism. This is where oxidative phosphorylation happens, meaning we use oxygen to help attach, or need oxygen to help attach, a phosphate group to ADP to form that ATP. Now, high energy electrons are going to be delivered here by either the NADH or the FADH2 molecules, which were produced earlier, either during glycolysis, if you remember, or the majority from the Krebs cycle, or in the case of using fats, beta oxidation as well. And so these electrons are passed down a series of carriers that are called cytochromes, like a step down staircase, losing a bit of energy at each step. And as the electrons move through the chain, the energy they release is going to be used to pump hydrogen ions from the mitochondrial matrix across the inner membrane into the intermembrane space. And this creates an electrochemical gradient where we have a stored form of potential energy, kind of like water building up behind a dam. And eventually the protons, the hydrogen protons, will flow back down the gradient through a protein called ATP synthase. And this is essentially the turbine that uses the flow of protons to power the phosphorylation of ADP into ATP. Now, oxygen is the final electron acceptor at the end of the chain, so it combines with the electrons and hydrogen ions to form metabolic water, which is why oxygen is essential for aerobic metabolism. Without oxygen, the entire chain backs up and ATP production grinds to a halt. So without oxygen, aerobic ATP production can't actually take place. And now the ATP yield is really where we see this massive payoff for the extended process that took so much time. So each NADH molecule yields about 2.5 ATP, and each FADH2 molecule yields about 1.5 ATP. And the difference is because FADH2 enters the chain at complex 2, which is a little further down the chain, and pumps fewer protons across the membrane. So when we put it all together, you have that one single glucose molecule that we started with. We get the 2 ATP directly from glycolysis, about 5 ATP from the NADH molecules produced in glycolysis. Then we have the 2 ATP from the Krebs cycle, plus the NADH FADH2 from the Krebs cycle going to the electron transport chain, which comes out to be roughly 32 ATP per glucose molecule. I'll say a lot of times now, as more research is coming out, they more often give a range like 30 to 32 ATP. All right, and so what we just described on the last slide is known as the chemiosmotic hypothesis. And so as electrons are passed along the chain of cytochromes, the energy they release is used to pump hydrogen ions from the mitochondrial matrix to the intermembrane space, which we see right here. Now, this builds up an electrochemical gradient, meaning that there's now more protons outside the inner membrane than inside. And so, again, this is where the analogy water building up behind a dam could be used. So now we have all this stored energy. And then these protons can only get back into the mitochondrial matrix by flowing through this special enzyme complex called ATP synthase, which is this little purple image here that I'm kind of covering up. Uh, sorry. So as the protons flow back down the gradient, ATP synthase spins like a molecular turbine, using the energy to attach the phosphate group to ADP, forming ATP. And then at the very end of the chain, this is where oxygen combines with electrons and protons to form water. And again, this step is critical because without the oxygen to accept those electrons, the whole chain stops, the gradient collapses, and ATP production stops. So again, oxygen essential for sustaining life. It literally keeps this process running and us and our cells alive. All right, and then here we just have a table tallying the total ATP produced from one molecule of glucose, which we've already kind of went over, but I thought the visual would help. So we start off with the 2 ATP, the 2 net ATP produced through that substrate level phosphorylation during glycolysis. And if we were using the anaerobic processes, that's where it would stop too. But with aerobic ATP production, remember we had those 2 NADH molecules that we talked about in class Monday. And so those would be shuttled to the mitochondria, go through the electron transport or drop off the hydrogen atoms to the electron transport chain. And we know each NADH molecule is going to give us about 2.5 ATP. So we now have 7 ATP as a result. So 5 from the 2 NADH and 2 from the substrate level phosphorylation of glycolysis. Now, we also get 2 NADH when each pyruvate molecule, because remember, end result of glycolysis, 2 pyruvate molecules. So when each pyruvate is converted to acetyl-CoA, we get a single NADH molecule. So pyruvate, 2 pyruvates to 2 acetyl-CoAs equals 2 NADH molecules. I may have left that out. If you go back and look at the detailed citric acid cycle, you see that when we're talking about producing carbon dioxide at that first step, whenever that 3 carbon molecule pyruvate is converted to the 2 carbon acetyl-CoA molecule, we also have an NADH that is formed as well. And then we have the 2 GTP, which is translated to 2 ATP through the substrate phosphorylation of the Krebs cycle. And then we have our 6 NADH molecules that are going to form by going through the Krebs cycle 2 times for each pyruvate molecule. And then we have our 2 FADH molecules that are going to be formed during the 2 rounds in the Krebs cycle. And so when we add the 2.5 ATPs up for the NADH and the 1.5 for the FADH, we end up totaling approximately 32 ATP. And so the NADH and FADH ATP is going to be from oxidative phosphorylation. The GTP, which translates to the ATP, and the ATP from glycolysis are going to be substrate level phosphorylation. Now, I also went ahead and added in the common amino acid and fatty acid used to generate ATP as well. And I'm currently blocking it right now. I couldn't figure out how to move myself, so guess I'm getting old. Can't work technology. Anyway, so what you end up seeing is significantly less ATP that is going to be produced whenever we utilize amino acids. And then palmitic acid, again, we won't go through all the steps with beta oxidation again, but we see that we get significantly more ATP per palmitic acid, or per one molecule of palmitic acid compared to one molecule of glucose. Again, showing how our fat reserves are a great source of energy, but again, only during low intensity, long duration events. And lastly, we will finish up by looking at how aerobic and anaerobic systems work together during real world exercise situations. Right, so energy to perform exercise always comes from a combination of aerobic and anaerobic pathways. If you remember when we first started talking about the different energy systems, and we were looking at the phosphagen, the anaerobic glycolysis, and the oxidative systems, we were talking about how it's not a off on switch, right? We don't just shut one system on, turn the other one on. All three systems are active all the time. It's just the relative contribution that changes depending on the intensity and the duration of the activity. So for short explosive activities like your 1 to 3 second max effort lift or a short sprint, nearly all of that energy comes from the anaerobic phosphagen system. As the duration increases, maybe into that 10, probably further into 10, but maybe 20, 30-second range, the glycolytic anaerobic pathway starts to contribute significantly more. I guess at the start of 10 seconds, you'll see a significant increase in the anaerobic pathway. And then once you hit about 30 seconds, that's where the glycolytic pathway has now become the primary pathway for energy production. Now, once you reach a couple minutes, looking at about 2 to 3 minutes, the aerobic system becomes the major contributor. And for anything lasting several minutes to hours, like running, cycling, swimming, the aerobic system provides a lot of the energy, most of the energy, especially whenever it is at a low intensity. Like cross-country runners during the race, they should be at an intensity that's still relatively high, and there's a good balance. Once you start getting above, your elite 5K athletes that are, running it in under 15 minutes, uh, and getting into your 10Ks and half marathons, that's where you see uh, the aerobic system really, really starts to take over. Now, if you look at the chart over here on the right of the slide, you can see the contributions that vary kind of based off of sport. So you have your weightlifter at the very tip top, your 100-meter dash that's almost entirely anaerobic. Uh, Maybe for 100-meter sprinters where it's 9.8 seconds. Some of us right now, whew, might take me 20 seconds to get the 100 meters in. So in that case, probably would drop a little bit on that percentage chart. But so we see the marathon, on the other hand, is nearly 100% aerobic. So it's at a low enough intensity, although again, for real marathon runners, still pretty high intensity. It's about 88% of VO2 max. So it really comes down to the duration of that event as well. Because remember, intensity is the most important factor, but then the duration of the event's also going to play a role in the percentage of anaerobic versus aerobic systems. And so understanding these interactions is going to be crucial for designing conditioning programs for not just athletes. A lot of times we use sports as an example, but also for your regular everyday individual. And coaches can emphasize the correct energy system during training. So, for example, using interval training to target glycolysis might be better for a team sport athlete, whereas a steady state training might be better for building aerobic capacity for distance runners or cyclists. And so athletes are then prepared for the specific metabolic demands of their sport. Right? So I always like to use the example. A lot of basketball players run about 2.5 miles in any given game, but it's not the same as if you had them just run for, say, 14 minutes straight at a very low intensity. That's not the same mechanics, not the same energy system that they are using during their actual sport. So instead of running, your offensive lineman for 3 to 5 miles at a very low intensity, you can actually build up their aerobic capacity similar to that steady state exercise with repeat sprints and interval training, at least high enough for someone like an offensive lineman where they are still able to recover when on the sideline.